As I mentioned in the previous video, at the end of Krebs cycle, the glucose molecule is already completely oxidized to carbon dioxide, but most of the energy is still stored within NADH as well as FADH2 in the form of high energy electron. That's why it's very important to have oxidative phosphorylation to extract this energy from NADH as well as FADH2 in order to produce ATP. Oxidative phosphorylation involves electron transport chain as well as chemiosmosis, and it happens at the inner mitochondrial membrane, or we say cristae, which is shown in this picture here. This membrane inside, we call it the inner mitochondrial membrane or cristae. What do you need to learn about electron transport chain is that redox reaction keeps on happening throughout the whole chain. First, you must have the concept of redox reaction, which we say oxidation is loses electron or hydrogen, while reduction is to gain electron or hydrogen. Let's look at the components of electron transport chain. We have 4 protein complexes that cannot move, which is complex 1, complex 2, complex 3, as well as complex 4. And you must remember the name of each complex. Complex 1, its name is NADH dehydrogenase. It will accept electron from NADH. Complex 2, the name is succinate dehydrogenase, and it will accept the electron from FADH2. Why do we call it succinate? Because during Krebs cycle, the step from succinate to fumarate involves the formation of FADH2. That gives the name of complex 2 succinate dehydrogenase. Complex 3 is what we call cytochrome C reductase, and then complex 4 is cytochrome C oxidase. How do we remember which comes first? It's like redox, so reduction, reductase, and then only oxidase. This will help you to remember the sequence of complex 3 and 4. What about CoQ and cytochrome C? These two, they are the mobile electron carrier, which they will help to transfer the electron from complex 1 and 2 to complex 3, as well as from complex 3 to complex 4. They can move about in the phospholipid bilayer. Another name for CoQ is ubiquinone. Now, let's learn what happened during electron transport chain. NADH dehydrogenase, it will accept the electron from NADH. So NADH gives away its electron to complex 1, itself oxidized, become NAD plus. And this electron to complex 1, from complex 1 it will go to CoQ, and then complex 3, the cytochrome C reductase, and then cytochrome C, the mobile electron carrier, will take the electron from complex 3 to complex 4, which is the cytochrome C oxidase. And lastly, this electron will be given to the final electron acceptor, which is the oxygen, O2. It's very important to remember that oxygen molecule O2 is the final electron acceptor, because we breathe in oxygen molecules and not oxygen atom. What about the electron from FADH2? FADH2, from the name 2, you should know that FADH2 will give away its electron to complex 2 instead of complex 1. Then FADH2 will oxidize, become FAD. This electron from FADH2 will be given to complex 2, and then from complex 2 to CoQ, and then same road, it will go to complex 3, cytochrome C, complex 4, and eventually to the oxygen molecule, which is the final electron acceptor. When this oxygen molecule combines with the hydrogen inside the matrix as well as electron, it will then form water. It's very important to remember this equation as well as the role of oxygen, which is as the final electron acceptor. This is how the whole electron transport chain occurs, where each protein complex over here, they will undergo reduction and oxidation continuously when they accept and gives away the electron. The main concept that you must have here is that from CoQ to cytochrome C reductase, cytochrome C and cytochrome C oxidase, the electron transport is the same, but the starting is different. NADH gives its electron to complex 1, and from complex 1, it will directly go to CoQ. Whereas for FADH2, from the name 2, it gives its electron to complex 2 and then from complex 2 directly to CoQ. And then the following road is the same. Lastly, this electron will be given to the final electron acceptor, which is the oxygen molecule. When this electron is transferred from one complexes to another, it will release energy, and this energy is used to pump the hydrogen ions, or we call it the protons, from the mitochondrial matrix into the intermembrane space. It's very important to know from where the hydrogen is being pumped. It's from the mitochondrial matrix into the intermembrane space. Complex 1, complex 3 as well as complex 4, they will pump the hydrogen ions from mitochondrial matrix into the intermembrane space. How do they get this energy to pump? It's from the electron transport. During the electron transport, it will release energy. And this energy released from the electron transport will be used to pump the protons H+ from mitochondrial matrix into the intermembrane space. What you can see here is that the intermembrane space will accumulate proton. So it will create a high concentration of H+ in the intermembrane space, whereas in the mitochondrial matrix, the concentration of H+ is low. This difference is what we call the concentration gradient. Gradient, which in this case, it means difference. So, the difference in the concentration of H+ is what we call the proton gradient. Proton gradient is simply the concentration difference of H+ in the mitochondrial matrix as well as the intermembrane space. As you can see here, intermembrane space has higher H+ concentration, whereas in the mitochondrial matrix, the concentration of protons H+ is low. This is what we mean by proton gradient, or another name, we call it the proton motive force. And what will this proton gradient or proton motive force do? It is used to force this hydrogen back into the matrix down its concentration gradient, because we want to balance the concentration of H+ in the intermembrane space as well as mitochondrial matrix. This H+, it has to go back into the mitochondrial matrix. But how can it go back into the mitochondrial matrix? This whole inner mitochondrial membrane, it is impermeable to the protons. How can this H+ go back into the mitochondrial matrix then? It must through ATP synthase. This is the only way where the H+ can go back into the mitochondrial matrix. There is no other place in the inner mitochondrial membrane or crystal that allows the H+ to go through. ATP synthase is the only way that allowed the H+ to flow back into the mitochondrial matrix down its concentration gradient. By doing this, it can balance the concentration of protons in the intermembrane space as well as the mitochondrial matrix. And what happened here is that when the proton flows back into the mitochondrial matrix through ATP synthase, it will activate the ATP synthase. And this ATP synthase activated, it will catalyze the phosphorylation of ADP. So ADP accept inorganic phosphate, it will then form ATP. This is what we want during oxidative phosphorylation, is to produce ATP. The whole electron transport chain does not produce ATP. It only gives the energy to pump the H+ into the intermembrane space, so to create the proton gradient. And because having the proton gradient, having the difference in concentration of protons, that allows the proton to flow back into the mitochondrial matrix, which then activate the ATP synthase, and this helps to catalyze the formation of ATP. From one NADH, after oxidative phosphorylation, it produces 3 ATP, whereas from one FADH2, after oxidative phosphorylation, it can only produce 2 ATP. And in total, we can get 34 ATP through oxidative phosphorylation. Together with the 4 ATP from substrate level phosphorylation, we can get total 38 ATP for active cell.